everyone, my name is Anthony. Today for my oral presentation, part 3 of STEM 2, I would like to address two questions. Question 1, explain the mechanism in which the fungal toxin Perfidin A affect the retrieval of KDEL tag protein from the cis Golgi network back to the ER. First, what the endoplasmic reticulum is. It consists of cystin A and the space enclosed by the ER membrane called the lumen. The two type ER, we have rough ER, which is a ribosome synthesized polypeptide protein within the membrane and then transport to the ER lumen. This protein then travel to procreate membrane to the cell surface by using secretory vesicle of the Golgi apparatus. Smooth ER, by its name, had no ribosome, had no growing protein synthesis, but instead they synthesize steroid, detoxify drugs, metabolize carbohydrates, and calcium storage. Golgi apparatus, this consists of full stack of system A, which is flat sac similar to ER. They have two faces of the system A. The first one is this Golgi network, with the consistent network of tubule that located closest to the ER. They have vesicle protein and lipid derived from the ER, moving from the cis Golgi network to the trans Golgi network by anti-Golgi movement, and the movement backward considered retrograde movement. For trans Golgi network, they consist of network tubule as well. They help to transport vesicle of lipids and protein to endosome, lysosome, and plasma membrane. This is what the protein traffic inside the endomembrane system look like. On the left, we have the ER synthesized protein and lipids into vesicle and transfer those to the Golgi apparatus in the cis Golgi network and eventually to the cell surface of plasma membrane for exocytosis exist outside to the cell itself. This is considered the anti-growth transport. At the bottom, we have the retrograde transport, where endocytosis pinch off the cell surface and transfer those vesicles back to inside the cell, to early endosome, to late endosome, to lysosome, to the Golgi apparatus, and eventually to the ER. So how ER retain their protein, ER retention and retrieval tags? Well, each protein has a specific tag to target the protein to transfer vesicle that would be carried from one specific cellular location to another. The tag help to retain protein in ER because they retrieve the protein that left the ER to the Golgi apparatus. So pretty much any vesicle of protein from the Golgi apparatus push back to the ER in the retrograde movement, that we consider a tag. For example, we have one tag called the tripeptide sequence RXR, which stands for adenine, X-alkyl amino acids, X is any kind of amino acids. Another tag protein, KDEL, this stands for lysine, aspartic acids, glutamic acids, and leucine. Their job is to promote retention in the ER of the mammals. They have short C-terminal amino acid sequence. The job is to bind to transmembrane receptor facing the Golgi apparatus. That when the binding, they induce the conformational change and cause the receptor-like complex packed fully with vesicle of target protein to return back to the ER. Another protein called the disulfide isomerase and BIP to ER resident. The job is to process this KDEL sequence and promote the retention in the ER. The vesicle that I told when pushed back from the Golgi apparatus to the ER, that considered the COP1 coded vesicle they found in eukaryotic cells, and they also support in bidirectional transport between Golgi apparatus system by itself, composed mainly of COP1 protein of 7 subunits and the AIF factor, which stands for ADP ribosylation factors that occur as part of the ARF GDP complex because it is considered a TDP binding protein. Their job is to encounter guanine nucleotide exchange factors in the membrane of the new vesicle to exchange GDP for GTP. This conformational change in the ARF expose the hydrophobic end terminal attached to the lipid bilayer of the membrane, and this ARF also mediate the COP1 protein because they bind to the COP1 protein and start the formation and budding of new vesicle. Then the protein in the donor membrane trigger the hydrolysis of TTP and push back to the ARF GDP complex to release the co-protein and repeat the cycle again. So how fungal toxin Perfidin A works? This is the lactone antiviral produced by fungus Penicillium perfidinum. The job is to inhibit the retrieval of the KDEL tag protein from the cis Golgi network back to the ER by prevent the association of the COP1 protein code of the vesicle to the Golgi membrane. So how does it work specifically? Well, this Perfidin A inhibit the guanine nucleotide exchange factor function of the ARF extract earlier to produce ARF GDP from ARF GDP. They describe that mechanism. So the COP1 vesicle can be no longer pushed back from the CGN of the Golgi apparatus to the ER. So that has to work. Question 2, how low concentration of Botox can be used as treatment for enclosed muscle spasm? First, Clostridium botulinum is a bacteria that can be found anywhere in soil, fruit, vegetable, and in the air. They can grow in anaerobic environment to produce botulinum toxins. They are 7-man type. Botulism is disease when people ingest toxin that they eat food that's not well preserved. For Botox, what's that? It's a paralytic toxin produced by the bacterium Clostridium botulinum. Their commercial form is a toxin type A from the 7-man type. Their function is to enhance youthful appearance for people who try to look young, try to avoid plastic surgery, or reduce aging. This is the structure of how the botulinum toxin A look like. The toxins here is initially in the inactive form, and you can see the translocation domain facilitate uptake into the cell through endocytosis. We can also see the reduced environment and endosome cleave the disulfide bond, releasing the catalytic light chain into the cell. And you also see the heavy chain over there as well. So this is how the toxin A enter the neuron cell and activate it. On the left side, we have how the normal neuron transmitter look like. We have the snare protein, syntaxine, the SNAP25. These are essential proteins required for fusion of neurotransmitter contain vesicle with plasma membrane of the nerve cell. Once attached to a vesicle contact to muscle cell, it will release an electrical impulse, initiate muscle contraction. Um, and you also see that the once the toxin A enters to the cell, right, it cleaves the protein and inhibit the vesicular fusion with the neuron membrane and release the acetylcholine. You see the vesicle over there, they contain acetylcholine because the one it binds to the synaptic cleft of the muscle membrane that will initiate muscle contraction. But when toxin A enters the neuron, it will cleave the protein, so then the vesicle can no longer bind to synaptic cleft. So that's why muscle contraction is being blocked, which is very deadly and condition of the brain cell. Very, 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 very deadly. And that's how you use a low concentration of Botox for treatment of wrinkle and muscle spasm. While small concentration can induce local paralysis of specific region of the face, wrinkle diminish because the muscle can no longer contract under the skin. Remember the acetylcholine guy, the neurotransmitter, they've been blocked, so muscle contraction can no longer be initiated. Benefits: smoother skin, less wrinkles, but downside, it's not gonna last very long, only a few weeks to a few months. This is my reference.